Hello, Ganesh sir. How are you?
Hey, good morning, everyone.
Good
I'm good,
morning.
Sangeeta. How about you?
Okay, I'm fine, fine. Nice to see you.
I know.
Okay, for another
Same
2-3
here.
minutes, wait, we will start. Okay.
Not a problem.
Okay. Vijaya ma'am, am I audible?
Yes, madam. Shall I?
Yeah.
Yes,
Yeah.
madam. Audible.
Yes, yes, yes.
We'll wait, or shall I start, madam? We'll wait for 2 more minutes. Hello?
Ma'am, 35 you start.
Yes, madam.
Madam, we will start now. You can start the function.
Start, madam?
Yes.
Yeah. I'll start, madam.
Yes. Yes.
Hello?
Hello.
Yes, yes. Is a continuing intervention of. Hello?
Little bit louder, madam. Yeah.
Hello?
You are audible.
Yeah.
Start.
Yeah, yeah. Life is a continuing intervention of the inapplicable. This very saying of Erwin Chargaff is echoed through the dynamic gamut of biochemistry. Walking ahead with the steps of enormous encouragement, I wish you all a very good morning and emanate my gratitude for your presence. Before we scroll ahead of this conference, let me highlight some of the significances of what we refer to as conference. A conference is a celebration of knowledge. In the modern age of free access to information, a conference of this nature verily becomes the enabler to expand the nexus of knowledge, ideas, and results in finding solution to the unknown problems. Therefore, in the arena of life sciences, biochemistry has evolved as a topical ascent for the entire amblet of biopharmaceuticals. So, let us cherish every aspect of it without further ado. A very good morning to everyone who has joined us today for the conference. Before we begin, I request all the audience to kindly put off your videos and please stay on mute. Queries can be posted on chat box and they will be addressed at the end of the session. I Ivavi Prabha, Associate Professor, Department of Biochemistry, the Oxford College of Science, welcome you all for today's conference, BioVenture during COVID-19 pandemic through the lane of biopharma and academia. To welcome is to show honor. To welcome is to establish integrity. I invite Dr. Sangeeta Roy, Head, Department of Biochemistry, to deliver the welcome address.
Please inform Prakash to start
Thank you,
recording.
Prabha Madam. Am I audible?
Yes, ma'am.
Okay. Good morning, one and all present here. A true visionary is one who knows how to
Good morning,
navigate
Dr.
in the
Chitra.
dark.
Please speak to
The
Prakash
second wave
and
of
ask him to
COVID-19 pandemic
start.
forced us to shut down all the educational institutes across the country.
Okay.
But it could not
Okay,
stop our enthusiasm,
Once we
dreams,
start,
and
we can
hopes
start recording.
towards learning and sharing our knowledge. Webinars have become a new normal of students' life. It is now a platform not only to learn new aspects, others' perspective and latest information, but also a new way of networking. Department of Biochemistry, the Oxford College of Science, is always instrumental in nurturing students' dream and perspective and never let it down during the pandemic. Department has taken different new initiatives to provide positive vibes in their lives. On behalf of the Department of Biochemistry, I, Dr. Sangeeta Roy, Head of the Department, take this opportunity to extend a cheerful welcome to all the dignitaries, my colleagues, all the research scholars, alumni, and the students in this national webinar series on BioVenture during COVID pandemic through the lens of Biopharma and Academia 2021. It is a great honor for me to introduce and welcome today's first guest speaker, Dr. Ganesh Ramachandran, Associate Director at Biocon India Limited, Bangalore. Dr. Ganesh holds bachelor's degree in pharmaceutical sciences from University of Mumbai, a postgraduate degree in bioprocess technology from ICT Mumbai, and has completed his PhD in bioprocess technology from ICT, formerly known as UDCT Mumbai, in the year 2008. Having work experience in biopharma industries more than 15 years, Dr. Ganesh has started his career as a senior scientist at Biocon India Limited in 2007. From 2010 to 2016, he worked at USV Limited Mumbai as a senior scientist and currently leading and mentoring a team of 7 scientists involved in process development, scale up, validation and production support of recombinant and synthetic peptides in Biocon India Limited, Bangalore. As an additional qualification, he has successfully completed the course on intellectual property conducted by World Intellectual Property Organization, WIPO, Geneva, Switzerland, and securing 100% marks in the exam. Dr. Ganesh has 3 patents in his credit and presented and published many papers in conferences and journals respectively. Dr. Ganesh has received fellowship from GATE, GRF from DBT New Delhi, and research fellowship from Department of Atomic Energy New Delhi, and recognized as an examiner for MTech and MChem engineering students at ICT Mumbai. We are indeed very honored to have such an eminent personality amongst us. On behalf of the Department of Biochemistry, the Oxford College of Science, I extend my heartfelt welcome to you, sir. It gives me immense pleasure to welcome our beloved chairman, Sri SNVL Narasimha Raju, who is the constant support in all our activities. It is my pleasure to welcome Dr. Kavirshi R., Distinguished Principal of the Oxford College of Science. She is an enthusiastic leader and has been instrumental in promoting growth of the institutions in various ways. I welcome you, ma'am. It is my pleasure to welcome Professor Gayatri Sudhir, Vice Principal Administration IQAC Coordinator of the Oxford College of Science. With over 24 years of teaching experience in Oxford College of Science, Professor Gayatri Sudhir has been responsible for providing strategic direction in the growth of the college. Madam has always been mentoring, counseling, and motivating students all through their stay in Oxford. Professor Gayatri Sudhir is the chairperson and member of Board of Examination and Board of Studies of Electronics for Bangalore University. She is also a member for Board of Education and Board of Studies in various other institutions. I extend a warm welcome to you, ma'am. I take this opportunity to welcome Dr. Bharti A.S., Vice Principal and Dean of Academics, Professor and Head Department of Microbiology. Madam is a person with distinct vision and a fountainhead of illuminating ideas. She has completed post-graduation from University of Mysore and worked for her doctorate at CFTRI Mysore. Dr. Bharti has been awarded with seed money for young scientists in the year 2013 from Vision Group of Science and Technology, Government of Karnataka. She is also a member of Board of Education and Board of Studies in Bangalore Universities and many other institutions. I extend a warm welcome to you, ma'am. I take this opportunity to welcome all the deans, HODs, faculty, alumni, and the research scholars present here. Last but not the least, I extend my heartfelt welcome to all the super enthusiastic, vibrant students from different colleges, universities across India for whom this webinar we have arranged. Hope this event will be fruitful in your future endeavor. Thank you very much. Over to Ivai Prova, madam.
Thank you, ma'am. Let us all kindle a start with an auspicious touch using this virtual platform to light the lamp and seek blessings from the Almighty.
शत्रुबुद्धि विनाशाय दीपज्योतिर्नमोस्तुते दीपज्योतिर्नमोस्तुते शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः.
Thank you all. Now I request Professor Gayatri Sudhir, Vice Principal, Administration, to address the gathering. Ma'am,
you can start the session.
Good morning.
Walk into the
Science
nature
is a
that walks the
lifetime
back
mission.
home. Good morning to one and all
I'm
present
delighted
here.
to
I,
participate in
Shivani,
the
NSS
inauguration
Manager
of
of the
webinar
Student Council of the
BioVenture
Oxford
during
College of Business
COVID-19
Management.
pandemic.
Nature
I
works.
greet the
Nature
organizers,
is
the
honest.
Department of
Nature doesn't care
Biochemistry,
about
Dr.
the human
Sangeeta Roy and
existence
team,
or doesn't even
distinguished
care about the
speakers,
human emotions.
researchers, and
I
my dear
welcome you all
students.
for the wonderful
As we
session,
are
the
all
World
aware,
Environment
the
Day,
Oxford College
organizing
of
by
Science
NSS unit
always
of the Oxford College of Business Management. Before moving into the session, I would like to present the glimpse of our NSS department.
Is ma'am got disconnected?
Yes, ma'am.
It's like she's frozen.
Okay.
Please call her and inform. No, she won't be knowing.
Rajesh, 1 minute, Rajesh, sound is not coming. Can you play once again? Rajesh?
Sure, sir. Just give me a second.
For control option and play it again. Stop presenting.
She's left and she'll rejoin, I think.
Rajesh,
Yeah.
1 minute.
Yeah.
1 minute, Rajesh.
Yes, sir.
Am I? Can you all hear me?
Yeah, you are audible, ma'am. Yeah.
Yeah. Once again, good morning. Science is a lifetime mission. I'm delighted to participate in the inauguration of webinar BioVenture during the COVID-19 pandemic. I greet the organizers, the Department of Biochemistry, Dr. Sangeeta Roy and team, distinguished speakers, researchers, and my dear students. As we are all aware, the Oxford College of Science always tries to develop scientific temper among its stakeholders. In the present time, when we are coping up with pandemic that requires awareness at the individual level in order to protect the collective and face the challenge. It is not just wearing a mask or social distancing. There is a need of raising general awareness about the pandemic as well as healthcare and hygiene. In these lines, the Department of Biochemistry has envisioned the webinar BioVenture during COVID-19 pandemic. The theme of this webinar is to bring awareness about BioVenture during COVID pandemic. We have experts from across the field with us for 3 days sharing their thoughts, knowledge and ideas. We have range of topics starting from biopharmaceutical industry, clinical trials, bioengineered nanoparticles and COVID and its impact. The department has taken great initiative to reach Biopharma and Academia. During this difficult time, it is the need of the hour. I appreciate the Department of Biochemistry for organizing this webinar. I would like to conclude by encouraging delegates to participate with an increased number in all the sessions in this digital platform. I know technical glitches will be there here and there just now as discussed already, but we are improving and we are taking it as a challenge and we are utilizing the virtual platform at the utmost. So I request and I wish everyone to have a successful and safe, fruitful webinar. I would like to conclude by saying Jacob Zemeckis once remarked that is the essence of science as an impertinent question and you're on the way to pertinent answer. I'm sure this webinar will create a platform for both pertinent and impertinent questions leading to or starting a path to pertinent answer where we will be able to find this pandemic and we will come back to that. I thank the organizers for giving me an opportunity and I wish all the best for 2020. Let us have a fruitful day. Thank you all.
Ivavi Prabha madam. Ivavi Prabha madam. Is it audible?
Ma'am, ma'am is in the list, but it's not replying.
Thank you, ma'am, for your enthusiastic and encouraging words. Yes, madam. Yes, madam.
Yeah. Carry on, carry on.
Hello.
Carry on, ma'am. Carry
Hello?
on. It's okay. Carry on, madam.
Hello. Yeah. Thank you. Thank you, ma'am, for your enthusiastic and encouraging words. May I invite Dr. Bharti S., Vice Principal, Dean Academics, to address the gathering. Ma'am, over to you.
A very good morning to all of you, distinguished resource persons present here. A very happy moment. I congratulate the Biochemistry Department for taking this initiative at a right time. This is most required, as Dr. Sangeeta mentioned. Webinars have become our new normal now because one cannot stop educating self and educating others, because this is the need of the hour, I feel. Because we have seen with this unwanted guest in the humankind, all are panicked. All are panicked without knowing what to do, what not to do. So if we go outside for, say, for our own vaccination or anything, we can understand and we can be really astonished to know what are all the understanding in the common man about this pandemic, about this vaccination, about this infection. Some end up saying that there is nothing called COVID at all. It is all political. Some end up telling that it will not infect them alone. I don't know. I really appreciate their confidence. No, it will not come for us. So these are the different things, and some say that vaccination is simply a political move. And I myself experienced all these things when I went out for vaccinating myself and my family and all. So I think educating ourselves and educating others is absolute necessary at this time. And future science, as one of the resource person is talking about, and I'm very proud to say that future science will be biological science, wherein people have understood the importance, especially, I'm sorry to say, I don't know the mindset in South. It's only doctor and engineering are the two careers which were always backed up, and they know how important is research in one's life, what is the important role the scientists play in this world. And I'm happy about the realization, but at the same time, our responsibility on our shoulders is becoming more and more day by day. So I really congratulate the team of biochemistry department who has planned to arrange for all these webinar series with various topics, and educating all our students and their related community. It is very nice that it is going on in the right time, and I wish the webinar all success, and I wish the students to take this seriously and participate in huge number and make the most of it. Thank you. All the best.
Thank you, ma'am, for your motivating words. It is always a pleasure to listen to your inspiring words. I am delighted to have Dr. Ganesh Ramachandran amidst us, and I now request you to take over the virtual platform. Our participants and students are eagerly waiting for your talk. Over to you, sir.
Yeah, thank you so much for the wonderful virtual inauguration of the webinar and all the talks by the distinguished faculty members of Oxford Science. Thank you very much for the wonderful introduction, Dr. Sangeeta. I just want to ask, what is the time frame I have, since it's 10:55 now?
Definitely 45 minutes to 1 hour.
Okay, fine, fair enough. I'll try to wind up by then. I'll just share my screen. So.
Okay.
Yeah, can you see the screen?
We can see you. You need to share the screen.
Yeah, I've shared the screen here. One sec.
Space. The world is struggling with the unprecedented pandemic. The pandemic COVID-19 confirmed the disastrous consequences of ecosystem loss.
Sorry.
Ideal conditions for
shall I just leave
pathogens
and
like
join again and check? I mean, it's not uploading.
And spread are created by
Prakash, system admin Prakash, is it okay?
Space natural habitat for animals and other organisms.
Yeah, give me a
Health
moment.
crisis of COVID-19 has compelled many of us to rethink our relationship with natural ecosystem and changing biodiversity. Interdependence between humanity and biodiversity is very profound. The hard fact is that without
Prakash, Ganesh sir has joined. Dr. Ganesh has joined. System admin Prakash. Dr. Ganesh has joined, rejoined. Okay.
Fine. Because not this from global that's far now here with the of here money okay we entire community that's and session.
Yeah, sorry for the delay, Dr. Sangeeta. For some reason, I'm unable to share the screen. Can you just present your screen because I've mailed the presentation? Will that be okay?
Moment, give me a moment. Let me take it from there.
So what they plan
is it visible?
Yes. Can you please go to the presentation mode?
Yeah. Is it okay? Ganesh, the PPTs
you can
are
go
visible?
to that full screen mode, Dr. Sangeeta.
Okay.
The lower icon on the left, right.
Sure. I will do. Is it now okay?
Just a minute. I think it's loading. Yeah.
Okay.
Just one slide back. Yeah. Thank you so much. Apologies from my side. Again, these technical glitches at the last minute. I mean, you don't know what happens when. So sorry for the delay. Very good morning to all of you. I mean, it's an honor talking in front of close to 240 students and faculty. Thanks to the organizing committee of Oxford College of Science for giving me an opportunity to present in this webinar. As a matter of fact, I mean, I have been passing through this Oxford College almost every day for the past 5-6 years en route Biocon. I mean, I was always impressed with the large campus, and it's really an honor to be talking to the faculty and the students of this university on this first day of the webinar. Thank you so much. So having said that, we will go to the topic, the first talk of the day, wherein since I understand the major audiences, bachelor's and master's students of biochemistry, I just want to give you a brief glimpse of what potential do you students have in the biopharmaceutical industry. So during the course of this presentation, I'll just kind of explain what caliber or what potential we have in the biopharma industry. So this is a disclaimer slide which we have to kind of publish as part of our organization, which says the opinions expressed are solely my own and it's not related to any of the organization that I am or I was associated with. Next slide, please. Yeah. So going to the brief agenda, I mean, over this next 45 minutes to 1 hour, I would like to just touch upon briefly the introduction of Biocon, along with a difference between what is a difference between a pharmaceutical and a biopharmaceutical. What are the career opportunities for biochemists? What are the role of biochemists in the biopharma industry? Along with the concluding remarks. You are free to stop me in between for any questions or any doubts, or else end of the session also we can have a question and answer session. So this gives us a brief update of what is Biocon. Biocon being in Bangalore, I mean, this was the first biotech company in India. It started off in the year 1978 as an enzymes company in a very small garage by our MD, Dr. Kiran Mazumdar Shaw, wherein it kind of transformed into a huge biopharma company between the year 2000 to 2004, where we went public, and Biocon was the first biopharma industry to be listed in the stock exchange. That's when Biocon divested the enzymes business and kind of focused mainly on both biotech as biopharma and generics. We also had a kind of partnership with some multinational companies like Mylan, Sandoz for commercialization of these biotech molecules across the world to kind of allow it to be afforded for all the public in the world at a very low cost. Biocon continues to invest in complex generic formulations. Also, it kind of has a lot of tie-ups with multinational companies for the commercialization of biopharma molecules. The pictures shown here is one of our production plants in Biocon Park, that is near Academi, where we manufacture most of our biosimilars for India as well as for the rest of the world. With a brief update on Biocon, I'll just jump on to the topic. Next slide, please. Yeah. So what is the difference between a pharmaceutical and a biopharmaceutical?
That was the key presentation
So
on how we have taken the earth for granted and how we have to
on the
live
left,
again.
pharmaceuticals
Thank you
are
so
mainly
much,
molecules
now we have a very
that are
interesting
generated through
I
a
hope
synthetic
everyone
route,
would be pleased
wherein
to
you
see.
just kind
Let's have the video, please.
put in some chemical synthetic reactions and your drug of interest or molecule of interest used in the treatment of diseases are formed. That is a pharmaceutical. Coming to biopharmaceutical, the process involved is huge. I mean, it involves with a genetic blueprint, that is a recombinant DNA technology, wherein you modify the DNA of the molecule, insert it into cells. Either it can be E. coli or CHO cells or insect cells, kind of generate the protein or the biopharmaceutical in large numbers, wherein you do a fermentation, harvest it, purify it to more than 99.8% purity because it has to be injected into humans. and then kind of it undergoes series of processing steps wherein you end up with a final drug, which is a biological medicine or commonly called as a biopharmaceutical. So put in simple terms, if you understand, pharmaceuticals are chemically synthesized, whereas biopharmaceuticals are grown, because it involves live organisms wherein fermentation and a lot of other unit operations are carried out to generate the final drug out of a biopharmaceutical.
Nature is sending us a clear message.
So this slide gives us a background of how the pharma versus a biopharma molecule looks in terms of size. So if you look at the extreme left of the graph, which shows aspirin, which is one of the oldest pharmaceutical molecule, which is being used even till today, wherein the size is very less, it's just around less than 100 Daltons. So all the molecules where you have a very small size, those are all classified as pharmaceutical. Once it crosses this size, it goes to a higher molecular size, that's when you start calling it as a biopharmaceutical. Again, you have segregations in that, wherein if it's less than say 50 amino acids or something, it's classified as a peptide. Once it crosses more than that, it is almost a monoclonal antibody or a protein. So all the entire bucket comes under one name, which is called as the biopharmaceutical. So in terms of examples, you see insulin is a very common biopharmaceutical, which is classified as a peptide. Other molecules wherein you see human growth hormone or erythropoietin or GCSF or monoclonal antibodies being used in cancer, anti-cancer, multiple sclerosis and all those, they are all large size molecules or biopharmaceuticals. So coming to the comparison, like exactly if you want to put down the critical quality attributes of a pharmaceutical versus a biopharmaceutical, what are the major differences you see? With respect to source, as I told you earlier, pharmaceuticals are generated through a chemical synthetic route, whereas biopharmaceuticals are through living organisms, that is predominantly a fermentation route. The active ingredients in terms of pharmaceuticals are very unique and well defined because the molecular structure is very small. So it's very easy to characterize that and define it or compare it with the innovator molecule. That is not the case with the biopharmaceutical, where the challenge is kind of built up. It is a complex mixture of too many heterogeneous proteins, and establishing a comparison between the innovator molecule and the biosimilar is really challenging. Characterization, pharmaceutical being a very small molecule, you have well-defined structures available, you have methods available in the pharmacopoeia, so it's easy to compare your pharmaceuticals. Whereas in terms of biopharmaceuticals, since the molecule is huge, there are challenges in characterization, so you have state-of-art techniques nowadays which kind of compare your biopharmaceuticals, but nevertheless it is much more challenging than the pharmaceutical characterization.
ಉಳಿಸುವ ಮತ್ತು ಬೆಳೆಸುವಂತಹ ಒಂದು ದೊಡ್ಡ ಜವಾಬ್ದಾರಿಯನ್ನ ನಾವೆಲ್ಲ ಮಾಡಬೇಕಾಗಿದೆ. ವಿಜ್ಞಾನಿಗಳು ಹೇಳುವ ಪ್ರಕಾರ ಭೂಮಿಯ ಮೇಲೆ ಈಗಾಗಲೇ 33% ಭೂಮಿಯ ಸವಕಳಿ ಆಗ್ತಿದೆ. ವಿಜ್ಞಾನಿಗಳು ಎಚ್ಚರಿಕೆಯನ್ನು ನೀಡಿದ್ದಾರೆ.
Coming on to immunogenicity, immunogenicity is kind of very rare in pharmaceuticals because it is a very small molecule, a synthetic molecule, so it's not known to be immunogenic. Whereas that is not the case with the biopharmaceutical. Since it is a protein with a molecular size of more than a couple of thousand Daltons, there is a large potential of immunogenicity being developed with biopharmaceutical. Again, coming to the manufacturing process, pharmaceuticals are much more easier to synthesize or manufacture, and they are highly reproducible. So you take multiple batches of pharmaceutical, it's highly reproducible. Whereas that is not the case with a biopharmaceutical, mainly because it is kind of a living organism, so it is sensitive to minor changes, and each batch is kind of very difficult to make and difficult to kind of mimic the exact manufacturing process in all the batches. So this gives us a very brief background of what differences pharmaceutical versus biopharmaceutical are. So coming to global scenario for biosimilar. So I'm moving in from a biopharmaceutical to biosimilar. I know there are a couple of doubts arising in the students' mind, like what is biosimilar? I have explaining it in the subsequent slides. Children, just keep aside for a moment that biosimilar is a biopharmaceutical. This slide just gives us an idea of how many countries across the world are working on biosimilars. You have a lot of regulated countries like US, EU, Australia, Brazil, and all investing a lot on biosimilar, with India also leading the forefront, wherein we have close to 42 biosimilars either being approved or in the pipeline. So this gives us an idea that what kind of potential you students have to contribute for biopharmaceutical research in the future. So we definitely have a huge potential to contribute in R&D for biopharmaceuticals. So just coming to a kind of a brief timeline as to how long does it take to generate biosimilar development. So biosimilar development can take close to 6 to 8 years to kind of start the R&D and end up with the final molecule. Why does it take so much time? So first 1 to 1.5 years goes away in kind of developing the clone which the innovator molecule has made. Other couple of 2 to 3 years goes in your process development, your process optimization of fermentation, your purification, your manufacturing scale up. After which a substantial amount of time goes in establishing the comparability with non-clinical studies as well as clinical studies. I presume the next speaker for the day, Mr. Somnath Chatterjee, is going to give us an idea of the clinical trials, where you students will understand what clinical trials are exactly about. But in a broad level, to develop a biosimilar is not a joke. It takes close to 6 to 8 years, where it involves cumulative efforts of a lot of people from different cross-functional teams, wherein biochemistry students, either with a bachelor's or a master's degree, also play a very vital part in this whole development of biosimilar. So as I told you, there are quite a few doubts that arise in our mind, like what is a biosimilar? How different is a biosimilar from a generic molecule as we address it in pharmaceutical? How are they named? Okay, and are biosimilars safe? Are they less expensive like generics? So do you get biosimilars for a lower price? And also, how do you compare that your biosimilar molecule is kind of comparable to the innovator molecule? I will try and explain most of these questions in the subsequent slides and kind of give some clarity to all of you. So coming to the definition of biosimilars, different kind of medicine agencies, like be it the EMA, that's the European Medicine Agency, or the Food Drug and Administration of US, or PMDA of Japan, they have different terminologies for biosimilars, but the bottom line for all of these is the same. So if you just take one example, say from the one in the first, that is the definition of biosimilar by EMA. So it says biosimilar is a biological medicine which is highly similar to another already approved biological medicine, which is a reference medicine. So when you say reference medicine, this was the first time that kind of molecule was made by one pharmaceutical company or biopharmaceutical company, which is known as the reference medicine or the reference. Are you there in this session? So other companies try and make copies of that reference medicine, okay, which is having properties exactly similar to that of the reference medicine. And they have to be kind of meeting all the standards of pharmaceutical quality, safety, efficacy that apply to all the biological medicines for that matter. So in terms, biosimilar is kind of a copycat version of a biological drug that was first made by a biopharmaceutical company. So similar kind of definitions are quoted by FDA or PMDA, wherein it says that whatever biosimilar molecule you make, it has to kind of match your innovator molecule. You don't say it is same because, you know, being such a huge molecule, there are going to be minor changes between your product and the innovator product. That's the reason it is called as a biosimilar. Similar means it is similar, close to, I would say, 99% plus to the innovator molecule. That's the reason it's called as a biosimilar. So I hope I have clarified what biosimilar means by the earlier definition. Going to a very layman language. Next slide, please. Yeah, I will try and give you a very simple example which should be able for you to kind of correlate what an innovator means versus what is a biosimilar. So I'm sure all of you in the summer times would have enjoyed eating mango pickles because mango is the season of summer. So most of us enjoy eating mangoes in different form. One more very famous use of mangoes is use of mangoes in pickle. So assuming most of us have eaten pickles made by our grandmother in childhood. So assume for a moment that the pickle, mango pickle made by your grandmother was an innovator molecule. Okay. Now, having stayed in the city, away from our villages, away from our hometowns, our mother kind of tries to make a similar mango pickle in the city. Now there are challenges in this because the mangoes that your grandmother procured in the village versus the mangoes that your mother tries to get it in the city, there are going to be slight changes in the quality of the mangoes. Similar thing goes along with the other raw materials added for making the mango pickle, which would involve all the masalas, the red chili powder or the oil. There are going to be some quality differences because whatever quality you get in your villages are much pure than what you get in your city. In city, you have some kind of adulteration. So you won't get the exact taste like what your grandmother made. So for a moment, you can consider that a innovator molecule is equivalent to the mango pickle made by your grandmother. A biosimilar is similar to the mango pickle made by your mother. Both are mango pickles, but yet there is going to be fine changes in the quality of the final mango pickle. That is still acceptable because we can't kind of wait for the mango pickle from our grandmother to come, or possibly grandmother is no more. There are several causes. Again, in this COVID time, there is no reason why we can get the mango pickles through a grandmother. So that's how a biosimilar and the innovator molecule kind of compare. I hope I have clarified like what biosimilar is with respect to innovator. Having said that, coming to the main topic, what career opportunities biochemists have in this biopharma industry? So biochemistry, as I told you, is the foundation for most of the biological processes. It is amalgamation of both biology as well as chemistry. So it helps us in giving kind of clues or answers for so many of the unanswered questions, be it in medicines, pharmaceutical, biopharmaceutical, forensic department, and also in the FMCG goods, like be it in cosmetics, foods. Biochemistry plays an important role in almost all these departments, all these industries. So it shows that biochemistry is kind of the future. It has been giving answers to all these areas, more so that with the advent of biopharma picking up, we have a huge role to play in the future, kind of contributing for all of these industries. So coming to the opportunities for biochemists in the biopharma industry. So how does a biopharma industry look like? I mean, I'm sure as students, even I was not clear what exactly goes into different departments in an industry, leave alone a biopharma industry. So I just thought I will give you a brief glimpse of how a biopharma industry looks like and where are the potential openings for we students as biochemists. So for any industry, research and development is the engine. It kind of, we are constantly developing new products for the benefit of mankind. Innovation is kind of through and through. It's almost 24/7. You keep on innovating something new. and sell your products. So that area which brings in revenue to the organization, that is the sales and marketing department. So here again, not many people with background in science get into this, but yes, you have a new role of technical marketing that is coming up, wherein people with a brief idea of the whole process are also kind of getting jobs in technical marketing and technical sales. So we as biochemists, we also have opening or opportunity in this department as well. So having said that, next important department that comes into picture is the regulatory affairs. So what definitions we saw earlier, like FDA has quoted this definition or EMA has quoted this definition. These are all regulatory bodies of that particular country, like US or Canada or Australia. So regulatory affairs department kind of help in selling or creating a benchmark for our molecule that is made in India in these regulated countries. So that is what regulatory affairs guy's job involves. You have to kind of meet all the regulatory guidelines of different geographies to ensure that your product is easily approved and sold in those countries. As a matter of fact, many people in regulatory affairs department also come with a biochemistry background. So here also we have a huge potential. Only thing we'll have to remember here is the kind of work profile involved here is more of documentation, where you study the different regulations and create roadways to understand how to go about getting your molecule approved in a regulated market. Last but not the least is the intellectual property division, which is kind of clubbed with the legal, wherein the people give a go ahead or say that whatever process adopted for manufacturing of this biopharmaceutical is free to operate in different countries and there are no IP challenges involved. They are also involved in kind of granting patents to the processes or products that R&D develops. So again, this involves a lot of studying of the available patents of that particular molecule, comparing it with the process or product developed in-house and giving a go-ahead. As a matter of fact, biochemists also have an opening or a probability of getting a job in IP. So if you look at this, I said I'll come back to the R&D division. So in R&D of a biopharma molecule, it starts with cloning, wherein you do genetic modification. There is gene modification, development of clones. That is one department. Once the best clones are made, it goes to the upstream process development, where people kind of do screening in shake flasks, kind of what we did in part of our microbiology work. And then you select the best conditions for your fermentation and scale it up to a fermentation level. That's what upstream process development guy does, followed by a downstream process group. Downstream process group kind of is involved in different unit operations of chromatography, crystallization, lyophilization, to kind of purify your biopharmaceutical molecule and make it up to the mark, what is desired by the regulated authorities. That's a brief idea of what downstream guy does, followed by analytical. So analytical is the method development that is done in R&D before it goes to the quality control. Last but not the least, you have two important groups which are involved in the final characterization, also the bioanalytical development of the molecule. So when I say characterization, we had said that you have to establish that your biosimilar is similar to the innovator molecule. So you establish it by different techniques available in the market, like mass or peptide mapping. There are several techniques. So that is what basically a characterization guy does. Also by establishing that the bioassays and all that of the molecules are well much controlled or comparable to the innovator molecule. So wherever you are seeing a green mark in this slide, we students as biochemistry background, we have a potential of getting jobs in all of these areas. So this shows that biochemistry is a very important area for biopharma market, and the potential is huge. So just in detail, like in R&D, if we are just kind of focusing on research and development, what are the different groups that are available and how the flow goes. So as I told earlier, first group involved is in the cloning, wherein you kind of clone the molecule and kind of establish new clones for your company to work upon. So that involves a lot of activities like gene isolation, your ligation, transformation, and clone finalization. This can be in different organisms, be it E. coli, yeast like Saccharomyces or Pichia. That again depends on what the innovator molecule is based on. So this is kind of the benchmark. It is a starting point for any biopharma research. Once this is done, the selected clones are sent to the upstream process development, where, as I mentioned to you, people work on the media screening and understand which kind of media or fermentation conditions give you the best output in terms of maximum biosimilar being fermented in a short duration of time. So there, apart from media screening and fermentation optimization, the guys are also involved in scale up from a 500 ml shake flask to a 2 liter or a 10 liter fermenter. This is the job profile that is involved in the upstream process development. Also, with the regulations in place, you have design of experiments, that is quality by design, wherein you use different DOE models to study multiple interactions of different factors for upstream or downstream. So that is also a very important part in upstream process development. Once the fermentation is done, the fermentation broth goes to the downstream processing group, where the team works on different purification strategies to kind of purify or isolate that particular biosimilar from other contaminants in the fermentation broth and take it to the quality that is desired, which I said is close to 99 plus purity. So again, the kind of work involves is more of process optimization along with DOE studies. The next important team that works on is all the analytical method development involved either in upstream, that is kind of supporting to your upstream process development as well as your downstream process development. So that can involve your enzyme assays, your SDS page, your Western blotting, characterization. You will have Edmond degradation, HPLC method, size exclusion methods, and multiple. I've just listed a few important ones, but this involves much more than these. The last two important groups that I mentioned was your bioanalytical method development, which predominantly works on bioassay development, radioimmunoassays, PKPD studies, and ELISA. And characterization is kind of comparing it that your molecule is very much comparable to the innovator, where high-end techniques are used to kind of establish the comparability, which can involve NMR, your high-resolution mass spec, FTIR, capillary isoelectrofocusing, circular diachroism, and so on. So this just gives a background like how many groups or how much potential is available for freshers to get into either of these departments. So having said that, I would like to just touch upon one important aspect. I'm sure most of you would be in your final year of MSc or BSc, wherein eventually after this COVID or something, we are going to start giving interviews and try to get placed in companies. So I would like to just touch upon how to be corporate ready. I mean, how is that we as freshers, we don't have experience in giving interviews, how to bag that job immediately as seamlessly as possible. I'll just kind of give you a few tips so that it can be of use when you go for the interviews post your graduation or post graduation. One thing is you have to have a resume, obviously. Don't keep a resume too long. I mean, I have seen resumes of entry-level people which are like 3 pages, 4 pages long. One thing important here is when a recruiting manager in any company screens your resume, he has just got a fraction of a minute to screen resumes from a bunch of resumes. So it has to be as crisp as possible so that first look at the resume and you are kind of shortlisted for the next round. So how do you do it? Do not give any do not hide or give any wrong information. Give all the information in a very crisp, short manner. You should highlight your achievements, but no need of giving details for all the things maybe that you did in school or your first year of BSc or something. Keep only the achievements in a very brief and chronological manner. I'm sure since you would have handled most of the techniques or instruments, it's always better that you give a brief summary of the project details along with what instruments you have handled. this kind of gives you a upper edge over other students, saying that, oh, you have handled maybe your UV spectrophotometer. So you have an edge over the other student or the other candidate who has not handled it. So that kind of sets your CV aside. Another important thing is to give references. But when you're giving your references, please inform your references. These references can obviously be your faculty members, but do inform them that you're giving their name as a reference, so that when the company does a background check, the faculty member knows that, okay, this was a student who had approached me and I am sure I need to give a positive reference about this candidate. Lastly, I would request, do a spell check on your resume, because I have seen many CVs of freshers coming with spelling errors. It is not aligned properly. These minor things matter when you're being shortlisted from a bunch of CVs. So kind of stick to that and keep a very crisp, flawless CV that is being sent out to the companies. Lastly, have a covering letter updated for each job that you're applying. You should not have one common covering letter for any job. So based on what job application comes in, you are supposed to kind of frame your covering letter in that particular way, tailor make it, kind of market yourself and attach it to the CV. That kind of gives you an edge of getting that job sooner. Lastly, you need to try, because applying for a job is kind of a job itself. So assuming we have given the CV in a similar way and you're possibly going to get an interview call. So before the interview call, how are you going to prepare for the interview? So with every interview, you kind of get a job description. So I would request that you analyze the job description and see what it mentions. It might mention several things of which you might have, say, around 60% or 70% of the skills. Focus on the 60-70% of skills and make a kind of pitching for yourself. You need to make a list of skills, why you are good at that, so that you can kind of make a pitch for yourself in the interview. Also, when you have a JD, you will have certain kind of qualities which they would have asked in the potential candidate. Kind of have your list of assets which can match with the ones that they have asked. Have real life examples ready, wherein you would have done some project work for your BSc or your MSc. Kind of have a example ready, wherein how did you go about the project execution? How did you go about project development? How did you face challenges? How did you overcome that? Prepare it backhand, so that because these are the common questions that people ask, so that you have something, at least 1 or 2 examples to tell during the interview. Next is check the company, what they are into, what are their products, how long they have been into this area. Check all those things. Also try to tap into your network to get the inside information, like what they are working on, so that during the interview, when they ask something about the company, you have a fair background of what the company is doing and what is it about. Lastly, as we generally do for any Viva or seminars, practice either in front of a mirror or practice amongst your friends. Ask your friends to ask questions and have an answer ready. Kind of polish yourself so that on the day of the interview, you do your best and you have already kind of practiced and it goes in a very smooth way. So assuming the interview call is arrived and you're being called for the interview. Nowadays, most of the interviews happen again through Microsoft Teams or Zoom. Nevertheless, once the COVID is over, you might have to have face-to-face interviews. For all of these tips are going to be useful, wherein have your interview clothes ready. No casuals, please, because for interview, you're supposed to be ready in formal clothes. Be professionally dressed. For girls, think about your makeup and accessories. No jazzy makeup or accessories, because you have to look professional. You have YouTube videos and a lot of articles available on Harvard Business Review, which kind of gives you how to get ready for interview. Go through that in your free time and prepare accordingly. Again, what to bring for a job interview, check with the HR team who is kind of coordinating with you. They might ask for your mark sheets or your reference letters or other credentials. Check it beforehand. If it's going to be a physical interview, exactly locate where that place is if needed. Try out how long does it take to go to that place. practice that. Again, if it's through some different medium, which is not Microsoft Teams, check it beforehand whether it is kind of seamlessly getting connected to that. Have a good body interview etiquette wherein your body language has to be good. You should not look slouchy, or you should look pretty confident, so that the interviewer kind of pays importance to all of these aspects, and all these kind of decide on what chances are there for you to get that the job. Lastly, however the interview has gone, it's always important that you follow up with a thank you note. Just after the interview, once you come back, send out a thank you note to the recruiting manager and the HR saying that thank you for giving an interview call, and also you can ask for any feedback if any. At times there are people who can give you a feedback which you can go on improving on for your subsequent interviews. Lastly, one thing I would like to mention is, if there are any questions asked wherein they say, give an example, a real lifetime example where you faced with a particular challenge and how you overcome this, have your responses as star responses. What does a star response mean? It says select a situation or a task that you had undergone, okay, actions that you took to overcome that particular situation or the task, and what was the final result. Giving this complete picture puts you in at a much better position wherein the whole story looks very convincing and the interviewer knows, okay, this is not kind of a built up, it is a real life story and this person has actually done something to get out of the problem. So all your problem solving skills, those things kind of matter in this, and that's how your chances of getting selected will improve. Next slide, please. So just to summarize, I mentioned to you what are the different groups or different departments in the biopharmaceutical industry. Apart from that, how to go and get that job in a biopharma industry, for which you need to do kind of a background research, have a crisp resume, do networking, practice your interview sessions. Once you get the job, you have to analyze your job, kind of tune your requirements to the particular skill set that they have asked for, prepare well for the interview, have a thank you note at the end of the interview. Once you have got that coveted job, it's always important that you keep your learning streak alive. It's not that once I've gone into corporate, you stop learning. When you are in R&D or something, you are supposed to keep your learning streak alive. Read, keep abreast about the recent happenings in your area. Also have integrity and punctuality. That's very important soft skills that is important for any candidate. Also have a curious mind. Be a team player, know how to work in a team, so that you will have to involve or interact with different cross-functional teams with different backgrounds. Networking plays a role in that. Work in a good team, embrace criticism. That's how you succeed in your corporate field. So very important thing is you have to have a right attitude, wherein connection between your attitude that you have today and the success that you'll get tomorrow is very important. So have a very good attitude, and I'm sure all of you are going to land up in good positions after your BSc or MSc. Lastly, to conclude, I would say education is our passport to the future. For tomorrow belongs to the people who prepare for it today. So keep preparing, put in your best, and all the very best for giving a listening ear. Thank you so much.
Thank you.
Now we open the session for question and answers.
Ganesh, I have a question.
Sure.
This is regarding this COVID pandemic. As our final year students are not getting that much of exposure, so what is the plan of the company? Are they going to recruit 2020 and 2021 batch? What is the plan for this company, to Biopharma company? Because you were telling different, how to prepare for the interview and others. But see the situation now. They have not written one semester. Definitely, they will write the final semester. What would be their future?
Yeah, it's a very good question, Dr. Sangeeta. One thing I would like to tell you is, I know COVID has affected us from last March, but as a matter of fact, Biopharma company is one of the companies which have been working through the last lockdown. I mean, as a matter of fact.
No, I must say Biopharma company is not in a loss. They are the company who are actually benefiting in this.
That's the reason I would like to tell you that the potential for hiring is still on, because we have a lot of openings coming up in our field wherein interviewing is happening through these online fields and people are being recruited. What I'm trying to say is, though COVID has affected different sectors of the industry, biopharma and pharma is one thing which is luckily not affected, mainly because we are into the development of either vaccines or drugs for this. So the job prospects look, I would say, bright for the students. Only thing is, since they have not given exams or something, students shouldn't be complacent. They have to be abreast, because eventually interview is going to happen. You're being kind of screened on the knowledge base that you have. So even though exams are not happening, students shouldn't be complacent. They should be kind of updated about the recent know-hows of the area and prepare well for the interview. Because with this online thing, I know starting from school students to college students, you have your 10th exams cancelled, 12th exams cancelled. One way it's good for the students, but that shouldn't make you complacent. Because eventually this COVID is going to end either next year or this year, after which the life is going to be normal.
Correct.
So definitely there is a bright future for the students, is what I would like to say.
Okay. Thank
you. Sir, there are some questions in the chat box. I would like to read it for you.
Sure.
This is from a master students, a biochemistry master students, Kavya. She's asking, sir, can you please throw some light on the role of genetic counselors?
Okay. I will just try to answer this because I'm not an expert in that field. But we had one genetic counselor during our master's course who kind of taught a couple of elective subjects. So I'll just try to correlate with that. So genetic counselors, I have seen, they kind of do genetic screening wherein you have some genetic disorders, say whatever genetic disorders you have, like retinitis pigmentosa or hemophilia. So wherein people kind of counsel the parents before they kind of go for planning for kids or something. So they are kind of associated with hospitals wherein they do some certain DNA extraction and compare what is the potential of the child being affected with that. I'm not very much aware of what are the hospitals that offer this, but I'm sure most of the hospitals which have a genetic department kind of is involved in this. I hope I could answer this, but I'm not really an expert in that field.
Thank you, sir. There's one more question from our guest, Soumya Murthy. Her question is, sir, could you please inform how MSc microbiology student can enter biopharma company?
Yeah, that's a very good question. If you look at the slide that I showed you, wherein different sections of R&D I had shown. So one important aspect there was your upstream process development. So upstream process development involves working with cultures or clones. So you do shake flask screening of cultures. That's where microbiology students fit in more. Though there are MSc biochemistry as well, but upstream processing is one area where microbiology students have an edge over the biochemistry students. How to get into that kind of industry is certain teams or certain companies, be it enzyme companies or biopharma companies, have these internship projects. It's like 2 months to 6 months internship projects post your MSc or as part of your MSc. You can kind of approach the company for that internship, do an internship there, and generally based on your performance during that internship and the availability of a position, people are hired. But that's the area where people fit in. That's the R&D part. In the production part, the same thing when it gets scaled up in a bigger fermenter, that is a 1 KL or a 30 KL fermenter. Again, the microbiology students are hired because you need to know how, you need to have a know-how of the intricacies of fermentation and how to culture, how to maintain that aseptic nature and all that. So that's where microbiology students can fit in.
Sir, there are a couple of questions. One of the student is asking, sir, HPLC, PCR, all this technical knowledge, is it very much must for having an interview preference?
I would not say you need to have everything, but again, it depends. Suppose the interview call comes from an analytical department wherein they are looking for a fresher who has to work on analytical method development, say enzyme assay development or kind of SDS page development, which we do it as a part of our MSc course or a BSc course. That's when being analytical department, the interviewer can ask you about the higher end instruments which can involve HPLC or GC. So even though you have not worked on it, it's always better to have a theoretical background of how the whole thing works, so that if any questions are thrown at you, you know something about it rather than saying I don't know anything. So you can always say that this is how the theory works, but I don't have any hands-on experience on that. So that will give you edge, saying that even though the student has not worked on it, he's aware of what it is or how it works. So it's better to know. But the same thing if it is for a different department. So that's what when I told you, you have to read the job description or the job profile correctly. So that's when you have to kind of fill in those gaps. So if it is for analytical and you don't know anything about HPLC, it's always better you read something about the analytical techniques and go for the interview. Same thing will apply for going for a downstream processing. You need to know something about purification techniques or other chromatographic techniques, so that you kind of are able to pitch yourself for that job and get that coveted job.
Sir, one more last question. Sir,
Sure.
could you suggest some coding languages which is useful to get into pharma company as a biochemist?
Coding languages.
What is the meaning
Yes,
of
sir.
coding languages?
Can she elaborate? I mean, is it related to IT she's mentioning? She or he?
So probably what I understood was the drug screening, something pharmacokinetics.
Okay. There is one, I think the University of Pune used to offer, I mean, this was way back in the year 2002-3, where one of my lab mates had done that 6 months course in molecular modeling, it used to be called, where you kind of work on the molecular modeling and kind of drug screening aspects. Apart from this, right now, to be honest, ma'am, I'm not aware of any other department or programs that offer this. I think we'll have to kind of search on the net where you should get more of the information.
Sir, one more question.
Sure.
Sir, are there any internship for undergraduate students which can help us get into this field?
Undergrad students, I don't think it is a part of any course curriculum as well, if I'm correct, because generally what I see is people who come in for internship are either from a MSc or an MTech, where you have to have 1 to 3 months or 6 months depending on the university as a part of internship, which is part of the curriculum. So if it is for a bachelor's, I think since it's not part of your curriculum, you need to take out your time and approach through your contacts during your vacation. That's when you can do a 1 month internship, and obviously it's going to add value to your CV. Definitely it will add.
Thank you, sir.
Over to Vijaya madam, over to Evadi Prova madam.
Yeah, yes, madam. Thank you, sir, for your inspiring talk, sharing your immense knowledge and precious time with us. It was absolutely wonderful listening to you, and I thank for your patient interaction with the students of the Oxford College of Science and all other participants. Your today's talk on science of future, biochemistry and its pivotal role in biopharmaceutical industry will certainly help us in our present and future studies. Thank you, sir.
Thank you so much.
Thank you, Ganesh.
Scrolling
Thank you for
ahead,
accepting our offer. Thank you.
Scrolling ahead, we are fortunate to our to have amidst us Mr. Somnath Mukherjee. He is presently working as Deputy Manager, Business Development, Syngene International Limited, Bangalore. A resourceful professional with more than 12 years of progressive experience and proven achievements in biopharmaceutical industry with in-depth insight, especially for large molecule bioanalytical as per regulated market requirement, along with techno-commercial experience for drug discovery activities in business development activities in the areas of safety assessment, in vivo pharmacology and research informatics. He has completed his Bachelor of Science in Biotechnology from University of Pune in 2005 and Masters in Biotechnology from the University of Burdwan in the year 2007. He has a technical expertise in ELISA, MSD, gamma counter. He has worked and interacted with the teams of global biopharmaceuticals from USA on multiple projects and presented data at their USA site. Sir is an excellent team player, identified as a go-to person for initiating projects, quickly grasps new routines and explanations, known to be self-motivated, results-driven, and able to work independently, makes sound decisions under pressure, confidently interacts with colleagues at all levels to lead implementations by delivering technical solutions and improving revenues, margins, and workplace productivity. In addition to the above, acquainted in a techno-commercial role for management of commercial proposals for drug discovery services, including safety assessment, in vivo pharmacology, and research informatics. We are very grateful to have such an eminent personality today amidst us. I extend my heartfelt gratitude and welcome you, sir. Sir, it's an honor and great pleasure that you are here with us today, and I now request you to take over the virtual platform. Over to you, sir.
Somnath, you can share your slide. Somnath? He was there only. He's calling me. Somnath, bol.
Yeah, I need.
Okay, just a minute. Prakash, he needs access to enter.
Yes, am I audible to everyone now?
Yes.
I apologize. There was a technical glitch, and I think my line got disconnected.
Yeah, it is visible.
Just a minute. Okay. So can everyone see my slide?
Yes, Somnath.
Okay. So thank you so much for giving me this platform to come across amongst you and discuss and share my thoughts on this topic of clinical trials. I thank Dr. Sangeeta for organizing this and inviting me for this talk. I was a part of Dr. Ganesh's discussion. It was really informative. It was really nice hearing Dr. Ganesh after such a long time. And I think the students are benefited with what he shared on the part of how to prepare yourself for getting an entry in the industry. So I chose this topic of clinical trials just to give you a briefing on what is a boom term right now across everywhere throughout the world. It's not that clinical trials suddenly came up in 2020 after the COVID pandemic, but it was existing very much in this world. Only thing is that common people were not aware of it. They were not so much informed about the work that goes in a clinical trial. So This is going to be an interactive session, so I'm not sure if I can.
Somnath, we can't hear you. Somnath, there is, your audio is.
Yes.
Prakash, system admin Prakash, is there any issue? His side only. Okay, I'm calling him.
Hello.
Somnath, your screen is visible, but your audio is not there.
Okay.
Check your connection.
Yeah, I changed my connection, and the audio is also on. Just let me try once again. Can you hear me now?
We can hear you.
Okay, I think there is some technical glitch out here happening. I apologize for that. So
No
just
issues,
let me know when you can't hear.
I will be informing you. You
Yes,
continue.
thank you
Just
so
first
much.
slide I'm
Yes.
seeing now.
Yes, correct. This is the first slide only that I was discussing, and this is of a medication that came up in the market during late 1950s and early 1960s. This is a drug manufactured by a company called Grunenthal, located in Germany, and they came up with this double thalidomide. So basically, this drug was being.
Should go to next slide.
This is the slide. Can you see my slide now on the thalidomide?
Only the first slide I'm seeing. The second slide should be there, no? That baby's photo.
Yes.
I'm not seeing.
Let me. Okay. Fair enough. I will reshare my screen. Maybe it has got struck. I'll reshare it so that you can see it once again. Just give me a moment. Are you now able to see the second slide?
No. Give some seconds. I'm not seeing. Your audio is visible, but there is nothing, no screen.
Okay, is it okay if I share my screen with share my presentation with any one of you, and you can control it from there?
Yeah, that's the same thing I did for Ganesh also. Some problem happened. Ganesh also unable to share. You mail me, Shobna. You know my mail ID, no?
Yes, I know your mail ID. Just give me a moment.
Mail me.
Yeah, just give me a moment. I'll share my
Biochemistry DPPT. I mailed you from there. You send me.
Yes.
I will download.
I will. Correct. Just a minute. I will just share you. Can you please tell me the email ID?
Biochemistry DEPT.
DEPT.
@theoxford.edu. It is there, your mail ID. If you check it, I mailed you from this email.
Let me see if it reaches you. Otherwise, I'll resend it.
I'm writing, Somnath. All in small.
Yes. Yeah. I typed everything in small. Okay. I have attached the PPT. Just let me know if you have received it at your end.
Not yet.
It will come. It will come.
No, Somnath, it is not coming. Something wrong?
It's in my draft and it should go to you. Okay, let me try from another mailbox.
Try.
Yes, I'm doing that. Okay, it has gone. It shows that it has reached the email ID that you have just sent to me.
Let me send in draft. Why it will go to?
It has gone to biochemistry department @the
Oxford.edu. DEPT, that's the problem.
DEPT. Yeah, it has gone to biochemistry DEPT @the Oxford.edu.
I'm not seeing there anything. Maybe. It has come. Give me a minute.
Okay.
Let me download. Give me a minute.
Sure.
Clinical trials overview, correct?
Yes, correct.
Give me a minute. It is a PDF, Somnath?
Yeah, it is a PDF. Okay, you can either open a PDF or yeah.
Can everybody see? Are you seeing my slides? Is it visible?
Yeah, we can see it. We can see it, Dr. Sangeeta.
Okay, fine. Somnath, you can start.
I think you can do Control L so that it will come on full screen.
Okay.
Okay, so I think there is some glitch here. I am unable to see the slide, but never mind. I will read through my slides and maybe I'll ask you to go to the next so that we are aligned and across the presentation. So, are you in slide number 2, Dr. Sangeeta?
Case study.
Okay. Yes. So I was talking about thalidomide. This is a kind of drug that was given to patients who are during the late 1950s and early 1960s in countries like USA and Europe, wherein people who have anxiety, sleeplessness, and stuff like that were being treated with this drug. Because of certain random usage, these drugs were suddenly started being given to people who are pregnant, in order to kill their morning sickness. So pregnant women generally have a vomiting sensation, and they generally generate it during the early hours of the day. So it was found that this medicine was somehow effective, although there was no testing being done actually on this medicine to treat this kind of clinical symptom. This was randomly given to a large number of population of people across Europe, and gradually, when these pregnant ladies started delivering, they found that their babies have not properly formed limbs. Okay, so their hands and legs were deformed, and this particular syndrome is known as phocomelia, wherein your hands and legs were retarded. They were not developed properly.
Yeah.
So this is one such incidence which raised an alarm across the world of of the importance of having a proper clinical trial. Dr. Sangeeta, can we go to the next slide? This is another case study which is for PRCA. Now, pure PRCA is pure red cell aplasia. This is also a kind, mostly it is a kind of genetic disease wherein your precursors for RBCs are not formed. So when your bone marrow starts forming the cells of that ultimately matures to RBC, there is a deformation in that generation stage, and these RBCs for these population, this cadre of people stops forming completely, and therefore they develop anemia. Okay, because RBCs are mostly carrying the heme part of the heme part, and that contributes to your to the body RBC content. So since the RBCs are deformed, they generate anemia. This was to a certain extent a genetic problem until in late 1980s. A Janssen drug started causing this disorder. So there was a drug from Janssen & Janssen which was known as Eprex. It was an erythropoietin analog which was given to patients being treated with cancer to stimulate their blood, blood count. Because during this treatment, there is a huge loss of blood cells. So in order to stimulate the blood production, they used to be treated with erythropoietin. Now, what used to happen is it was found across the world that 10% of population was found to have developed PRCA who has been treated with this particular manufactured product of erythropoietin. Upon further investigation, it was found that for a particular lot where this particular syndrome was available was seen, during the manufacture of erythropoietin, a stabilizer was used, which is known as polysorbate 80. It's a very common stabilizer. We call it in the industry as twin 20 and twin 80s. So this twin 80 was one of the component which was being added as a stabilizer to this drug, and that somehow reacted with the packing, the sealing of the vial, and that caused a modification in the drug property, which kind of stimulated the cause of PRCA in those patients. Why I gave you these two examples are that it is very important for a drug to be properly assessed before it is released in the market and being given to individuals. When I say assessed, I mean to say that the drug's safety and efficacy, both of these are like the magic words that you kind of hear all over the world, especially because of this COVID thing. So safety and efficacy both has to be assessed, and that is where the clinical trials come into picture. The next slide, please. So this slide actually talks to you about what exactly is a clinical trial. I have tried to put in, the theoretical definitions that are available in journals and textbooks. But in a nutshell, what a clinical trial is what exactly a clinical trial is, it is about a research that is aimed to, to see how fit a particular medicine or a surgical instrument being used is for its intended purpose. You actually kind of evaluate through a clinical trial how safe and how effective a particular drug or a medical device is. So when I say medical device, it means, pacemakers or catheters that are being used, they also need to be tested for its toxicology. May not be a part of directly clinical trial, but the primitive of clinical trial wherein the toxicology studies are being performed. So it is a holistic branch wherein you actually see whether that particular drug or that medical device is actually safe for its utility for human population. The next slide, please. So we all have heard about this phase 3 trials of these COVID vaccines that are currently running in. So I thought I'll give you a nutshell of how exactly this clinical trial goes through and what are the different stages in it and what each stage does. So before a drug comes to a clinical trial, there are a lot of discovery and preclinics that are being done. When I say discovery and preclinics, it takes almost 3 to 6 years to complete this entire phase before that particular molecule enters the phase 1 of clinical trial. What happens in this discovery and preclinics is that these drugs are being tested in animals, for example, rodents and higher non-rodents, which can include higher primates. When I say rodents, you do tests in rats, rats, mice, guinea pigs. And when I say higher species, it can be a dog or a mini pig or even monkeys. So these kind of testings are done to evaluate the toxicity of the drug. Okay, it is ethically not correct to administer a drug which is being worked out on a manufacturing level directly to a human population without having a basic idea for its safety and toxicity. So when I say toxicity, it means the harmfulness of the drug. So during these preclinics, what exactly is done is that you dose those animals across different different concentrations of the drug to see what is the maximum drug that can be dosed, which does not have a harmful effect or a toxic effect to the animals. Now, how you decide this toxic effect after the particular phase of drug administration is over, you euthanize the animals, you kill those animals, you take out different organs, and you try to see what impact those organs had because of the administration of the drug. So you can see a positive effect and you can see a negative effect. Now, these can be correlated to the efficacy of the drug. When I say efficacy, it means what the drug has done to the body. And when I say safety, I mean that what the body has done to the drug. So these are two very important concepts that we need to remember when we are trying to understand the clinical implication of a drug. So once everything is established on the preclinical platform, the study goes to a clinical trial phase. So there are different processes wherein you have to inform different regulatory bodies, get their approvals, and post that you have to start the phases. Now, a typical clinical trial earlier used to have 3 phases: phase 1, phase 2, and phase 3. I'll explain you what each of these phases are about. But now there is something called phase 4 as well. In my next slides, I will try to give you a hands-in about what each phases contribute, what are their differences, and how they help in identifying whether the drug is safe and effective in treating that particular indication. Next slide, please. So we have a phase 1 trial wherein the drug is being tested in healthy individuals. We have a phase 2 trial where the drug is tested on a mixed population. Some may have the same disease, some may not have the disease, but the population, the sample size increases. We have a phase 3 where this drug is being tested in disease population, and we have a lot of permutations and combinations included in this phase. When I say the permutations and combinations, it means the drugs can be randomized, the drugs can be double-blinded, and it just gets tested across different countries, to have a higher demographic access of the drug and see its impact. Post that, there is an FDA approval that is being sorted. When I say FDA approval, it is not only FDA, it depends on the regulatory body against which you are doing the job. As Dr. Ganesh has rightly said in this regulatory authorities, RA team of a company figures out what are the, what are the parameters that need to be tested for a particular clinical trial in order to go and submit that particular, clinical trial report to that to that particular region or authority. It becomes very important to, perform the trial based on the regulations being set up by that country. For example, if you are going for a submission of drug for US regulation, US market, you have to undergo the drug, you have to undergo, like, pass on the clinical trial as per the FDA regulation. If it is an European, if it's for European Union, it has to pass on through ANVISA. Japan has its own regulatory authorities, which has their set rules and regulations. And Visa, for example, is for Brazil. So there are many regulatory bodies with based on whose recommendation you have to actually go about and perform the clinical trial. Post FDA approval, it doesn't mean that the drug is ever safe. Okay, so you have to keep a watch on the drug, and that's where the phase 4 trial comes. This phase 4 trial essentially is to see safety, that this drug is safer for a longer duration, is what you ensure. And there is no timeline for phase 4. It can go up to years and years. The more the data, the more information you collect. So a phase 4 trial mostly is a continuous process, and it essentially targets on evaluating the safety of the drug, post its marketing. We'll go to the next slide, where I will try to tell you what each of these phase does. So a phase 1 trial is essentially when a new drug is administered for the first time on human beings. So whatever information you have collected from the animal studies, you translate that to a human study. You generally take a lesser population, maybe 10 to 12 individuals, volunteers who are healthy. Now, why you need to test your drug on a healthy individual? This is because in a healthy individual, you can actually assess the effect of the drug, because it will not have any other, ancillary parameters which will interfere on the efficacy and safety. So a healthy individual is, you can imagine, is a clean slate. So on a clean slate, if you strike with a blue chalk, you will see the blue color being visible amongst a white background. So similarly, that with that analogy, a healthy volunteer is being taken, so that whatever effect the individual is showing after the drug administration is only because of that drug, and not because of any other ancillary complication that the individual may have from the beginning. So it is very important to do this, that this is a high risk phase, but this phase actually gives you the base information with which you can proceed with phase 2 and phase 3. We'll see to the next slide, wherein I have kind of jotted down the key points, what you kind of get information from the phase 1 trial. And the main point out here is the safety. So what you evaluate is the safety of the drug. Okay. One more important thing out here is that placebos are pseudo drugs. Okay. These are drug mimics. You may call it like, your drug is a pink color tablet that is being given. You mimic a similar pink color structure, which is not a drug. It may have sugar, or it may have basic ingredients that you eat. It is just given randomly to patients, just to mimic the situation. I'll tell you more about placebos when we go to phase 3, because that is where the placebos come into picture. For now, you can keep a note that, for phase 1 trials, there is no placebos being given. So in a nutshell, what exactly is being done in phase 1 is that healthy volunteers are taken, smaller population is being tested. You basically see the safety of the medication that you are trying to bring it to the market through this phase 1 trial. And it lasts for like several months to a year. That's it is the span of a typical phase 1 trial. Now, once your drug passes phase 1 trial, it goes to phase 2. Most important thing to note out here is only 70% of the drugs which are tested in phase 1 moves to phase 2. So not all drugs make it to phase 2. These are kind of screenings, sieving the drug to find it which is most suitable for human consumption. Next slide. What does a phase 2 indicate you. So in phase 2, what you actually try to see is whether there is any clinical indication that is getting impacted because of administration of the drug. So I said, like in phase 2, you have a mixed population, few healthy and few disease. So what exactly you see in a disease population is that whether there is any reduction or any change in the clinical indication. For example, if you're giving a cancer drug, if a clinical trial is happening for a cancer drug, you tend to see whether the cancerous growth has been restricted, or whether it has shrunken in its size. So these are few indications that you try to see in a phase 2 clinical trial. This is mostly done in a larger population, just to ensure that if you have missed out some clinical indications in a smaller population, you have a statistically larger population subjected to the same kind of treatment, wherein it should be evident if there are any kind of safety concerns regarding that drug. The next slide actually talks to you about the key take-home messages for a clinical trial for phase 2. As I said, it is a larger population, so it means 25 to 100 patients of same disease type are being taken, so that you can see the efficacy of the drug, whether it is happening at all or not. You generally give them same dose. That means whatever doses that you have figured out during phase 1, you fix one dose, and in presence of the clinician, you kind of administer the same dose. Okay. Placebos again are not being used here. We'll move to the next one, which is the pivotal one, which is phase 3. Now, phase 3s are the ones which are more prolonged studies. So these, an ideal phase 3 may last from 1 to 4 years. And here is where we have these concepts of randomization, placebos, and double-blinded studies come into picture. I'll give you an example of each, and I'll try to explain you what these are. So in a phase 3 trial, you actually see the safety and efficacy both in parallel, because these trials are being done in disease population. For example, if you're doing a trial for a human insulin, which is being manufactured by company X against a already available drug, which is marketed, which was a novel few years back, and then a biosimilar came. You want to compare drug A versus drug B. So that is where your concept of randomization come into picture. So there may be considering two different groups. One group is treated with the reference item, which is already available in the market, which has proven results, and group B is treated with for example, the one that is being currently in test. After a certain duration, you do a crossover. So you switch the drugs, you make the reference item being treated for group 2, and the drug for group 2 moves to group 1. This way, you can actually evaluate the efficacy of both the drugs, whether they are actually impacting on the actual indication in the patient or not. Similarly, a double-blind study is that where both the doctor as well as the patients do not know what they are being treated with. So as per the clinical trial norms, the volunteers are not made aware of what they are being treated with. This is kept secret from them. This is as per the laws, in order to not let them know what they are being treated with. At some cases, if it is single blind study, the doctors know what they are treating, but in order to remove all possible kind of bias, the studies are mostly made as double blind, so that neither the doctor knows nor the patient knows, and the study becomes an unbiased study, and whatever results come are true to its intended purpose. Here also there is a concept called placebo comes into picture. A placebo is a pseudo drug. It is a drug look-like, but it is not made up of any active ingredients that a drug composes. It may be a sugar shot or you know a sugar coated stuff which is just given to a patient. Now this is done in order to see whether if at all the patient is basically a control group, you know, in order to see that, okay, with reference to a patient group which has a placebo, there is a change, if at all any, happening in the groups where the patients are being treated with actual drug. Now, there are certain instances where it was found that these patients psychologically also, you know, start curing themselves. So those psychological effects are being evaluated by these placebos. So when a patient who does not know whether he is getting a drug or a placebo is treated with a placebo, if the disease gets psychologically treated, that impact is also a part of a clinical trial study, and that also is evaluated, and that is done during a phase 3. So the phase 3 actually takes care of a lot of evaluations, and that is why these phases are basically a very prolonged phase study. We'll go to the next slide where I try to discuss on the key points of different, for the phase 3 trials, which is it includes a larger number of people and it can happen across different countries. So you have you might be knowing that, you know, each country has its people with a different genetic composition because of the environment and because of, the exposures that the individuals have got. They have a different genetic composition. Now, you have to evaluate that your drug is a robust drug, and that you can only do it once you see that your drug is equally effective across different races and different demographic regions of on the globe. Trials are not restricted to one region. They are generally being shared across many, just to ensure that, you know, the robustness of the drug is well established. These placebos, as I told, will be used, which helps in identifying a different set of people, which whether the treatment is actually happening psychologically or not. And this is what a phase 3 does. Now we'll go to the next slide, which talks about our phase 4 trial. Now, as I told you, after an approval, the drug comes to the market, and then the phase 4 trial starts. It actually tests the longevity, the safety of the drug in a long run. So there are random samples being taken up from population, in a larger population. This becomes a much larger population as compared to phase 3. And the samples are being taken and evaluated just to see whether there is a safety concern amongst the drugs that has been already marketed. We'll go to the next slide, which talks about the key points. So a phase 4 trial looks at the drugs that has already been approved by FDA. These involves thousands of people. As I said, the population side is high. And this is often the safest type of clinical trial because treatment has already been studied in lot, and it has been the phase 4 trial is the safest. Why? Because it has passed phase 1, phase 2, phase 3 and FDA approval, and then only is being tested. Now, the essence of this phase 4 is to see safety as well as whether there is a change in the quality of life. Now, this is also a very important aspect, whether after getting the drug, whether there is an impact on your life, whether the drug has helped you out in any particular kind of avenues that makes your life better. That studies are also, that observations are also being made a part of the report for a phase 4 clinical trial. The next slide actually talks to you about the typical workflow for a clinical trial. Now, these are generally work that are being done by clinical research associates, wherein they explain the volunteer why the trial is being conducted. Then they have a set of questionnaires which the volunteers are being asked to fill so that they have a proper knowledge. As well as having the knowledge, they give you a consent. Now, having a consent from the volunteer is very important because you cannot randomly pick up a person from the road and start doing a clinical trial on that. So there is a process, and it has to follow a particular procedure, which is being guided by ICH. ICH is a regulatory body, which is International Council for Harmonization of Technical Requirement for Pharmaceuticals for Human Use. So this particular regulatory body has set up a set of guidelines following which this workflow has been made. So after you have got the consent from the volunteer, you have to screen the volunteer. I told you, right? Because each of these clinical trials will have this, inclusion and exclusion criteria. You should have a specific body weight, a specific, origin, based on which the efficacy of the treatment will be evaluated. So all these inclusions and exclusion criteria are being evaluated, post which you are enrolled for a particular clinical trial. Now, once clinical trial is being done, these samples are being taken out at different intervals based on the time points being given in the clinical trial summary, and then it goes to the bioanalysis. So where this is the place where all these clinical trial samples reach for their efficacy and safety studies. So when I say safety studies, what they see is whether the drug has generated any antibodies in the human body against them. This is essentially a phenomena for large molecules and not for a small molecule, because immunogenicity is not a phenomena which is observed for small molecules. Only for large molecule, we have this immunogenicity aspect to be evaluated. And next is the efficacy. So efficacy is where you see whether certain markers that are present in the body has because of the administration of this drug. For example, you have a drug being manufactured which reduces cholesterol. Now, because these large molecules are very target specific, it goes and particularly binds to a particular receptor which kind of stimulates extra production of this, which actually hinders the metabolism of this cholesterol. So when I say efficacy, what you do is you go and see whether there is a reduction in the production of this particular biomarker. If there is a reduction, then only you can say that, okay, my drug is working, it is going and binding to that particular receptor, and it is not, it is blocking it so that the normal metabolism of the cholesterol happens, and there is a reduction in the cholesterol level. You do a lot of tests like LDL and VLDL testings, and then you see that, okay, my blood samples have shown reduction in the cholesterol level because of the treatment of drug. That means the drug is effective. Okay, this is how you actually assess the safety and efficacy. Now, efficacy is fine. You can directly correlate that, okay, I have administered the drug, there is a reduction in the cholesterol, and so my drug is working. Now, how do you know that? The moment you see that, although you're introducing a drug and the levels are not reducing, the first question that should come to you is whether there is an antibody being generated against the drug, whether there is a safety concern. Because if the antibody is generated against the drug, the antibodies will go and bind to the drug and it will not allow it to function for its intended purpose. Now, that concern is what you have to evaluate during safety. And once you have evaluated, okay, there are antibodies, the next step will be to find whether there are binding kind of antibodies or neutralizing kind of antibodies. I'll let you know about them as we progress through this presentation. Dr. Sangeeta, you can skip those 3 slides, the next 3 slides, and can directly go to slide number 19, which is for a biosimilar. So the slides that we skipped are the ones that talks about how a particular person can enroll in a clinical trial and what are the different stages through which the person has to go in order to call himself a volunteer, which is not required at this point of time. So I directly come to a concept called biosimilar. Now, this is a very boom in the market right now, and these are the drugs which are currently accelerating their usage in globally because of their target specificness. So biosimilars are basically biological products which mimics a similar product which is already available in the market, and these are essentially large molecules. Now, the advantage of a biosimilar, for example, an insulin, many people who are diabetic takes an insulin. Insulin is nothing but a biosimilar now. It's a large molecule. It's a large molecule component which mimics the structure of human insulin, but manufactured in a host, which may be a E. coli or a PKI, as Dr. Ganesh said. So there are different stages of manufacturing this insulin. You kind of clone the gene for expressing this insulin in a vector, and then you scale it up in upstream, and then you purify the insulin in a downstream, and then you characterize whether the properties are matching with the one that is available in the market, and then it goes for the clinical trial. So these are the different stages that drug manufacturing actually takes place. But the advantage of having a large molecule biosimilar is that they are very target specific. See, a small molecule can go and they can target the infected organs as well as the non-infected organs, because they are not target specific. But these molecules, biosimilars, are very target specific. They are targeted to a particular receptor or to a particular site. So that is the reason you see a huge amount of risk reduction for a biosimilar molecule. So the safety and efficacy, these vaccines which are being discussed now, is nothing but a biosimilar. So the concept of safety and efficacy during a clinical trial actually comes out because it is a biosimilar molecule. So we'll go to slide number 21, the stages that impact the variability. So as I said, a biosimilar molecule is a molecule which is manufactured taking a marketed drug as reference. So we cannot mimic 100%. There may be variations that may arise. Okay, now in a clinical trial, what we are evaluating is whether those variations are going to impact on the efficacy and safety of the drug as compared to the one which is already available in the market. Now, we will try to see where all these variations are coming from. So from your last presentation, you already saw that there are different stages of a drug manufacture. You have cloning, you have upstream, you have downstream, and in each stage there is a possibility that you are introducing a variation, and these variations on a degree of the variations that is being introduced, you will find the change in the safety and efficacy profiles of that particular drug when compared to a reference drug which is already available in the market. So the variability are introduced in the cloning stage, in the cell upstreaming stages, in the purification stages. It can be introduced in the formulation stages. So there are multiple places where a biosimilar can be subjected to a variation, because of which there is an impact on its safety and efficacy. The next slide talks about the factors that impact on which is impacting on the safety and efficacy. The first one is the post-translation modification. So once you have expressed a particular gene in a vector, you have to chop the leader and the Taylor sequence. When I say a leader and a Taylor sequence, it is the sequence with which the vector's translation and translation mechanism is used in order to get the protein product out of it. Now, after your final product is ready, you have to kind of chop it, and after the product is expressed, there are different post-translation modifications that are being done. These choppings, if they are not done correctly, and if there are portions of the vector's gene sequence or a proteins fragments which are present along with your protein of interest, that is where there is a concern on safety and efficacy of the drug rises. So this is one such major region where there is a huge chance of a concern on the biosimilarity from the reference product, and that is where the safety and efficacy comes into picture. Now, what happens when there is an alteration in the safety part? So that is where the next slide is talking about is immunogenicity. So immunogenicity is something which is unwanted for a biotherapeutic. Okay, you don't want your drug to be administered in the body and generate antibodies so that the drug's action is being nullified. You don't want to have that. So immunogenicity is unwanted characteristic that you want to see for a biosimilar. But counter to that, for a vaccine, what you actually see is, you intend to see is immunogenicity. So you have to understand that biosimilars and vaccines are different in their working. So when you inject a vaccine in the body, what you want to see is that it generates antibodies so that your body is well equipped to fight with that particular pathogen when it comes again and attacks your body. But for protein biotherapeutic, for example, a PEG-CSF or an insulin or a monoclonal antibody that is being used for therapeutic treatment, you don't want the antibodies to be generated. So based on these two concepts, the safety and efficacies are evaluated for a clinical trial. Now, the next slide actually talks about the factors that that induce immunogenicity. There are multiple. You have sequence variations, whether it's a humanized one and a bacterial one. So as I said that you are expressing it in a vector. It can be an E. coli or a picky, and when the protein is expressed out, it may have the at the flanking regions of the final protein product having sequences protein for the host. That has to be properly chopped out, and if it is not, either during your purification step or during your characterization step, if it is not being identified and chopped out, this particular product may prove to be immunogenic and may not be able to pass on the biosimilarity criteria. Then the amount of impurities. So in a downstream process, you as Dr. Ganesh said, you have to have a knowledge of different chromatographies. You should know about a size exclusion chromatography, ion exchange chromatography, affinity chromatography. So using all these concepts across different stages, a final protein product is being formed. Okay. Now, if these final product, after all these purification steps, if the final product is not pure enough and it has amount of impurities which can impact on the safety, then this will not again pass the biosimilarity criteria. So it is very important to have a check on the impurity profiling, whether while you are purifying the final product during this chromatography downstreaming stages, you are also eliminating chunks of impurities at each stage of the chromatography. That also has to be ensured. Then difference in the formulations, as I told you that, a particular batch of erythropoietin used polysorbate 80 and that changed its immunogenic profile. It raised antibodies against that particular drug, and then symptoms like PRCAs come into picture. So it is also important to select the right variant of formulation product. Route of administration. This is also very important, because if you are injecting a drug through IM or SC, the intramuscular or subcutaneous, your immunogenicity or the effect of response of body towards the drug also gets altered. The dosage, this is also very important, that amount of drug being administered. If it is administered extra, that drug can also be proven as immunogenic. Length of the treatment, the duration, like how long you are being administered with the drug. If there is too much accumulation of the drug in the body, then also it can cause immunogenicity. Patient's characteristics, how there are few, for example, like I just got vaccinated with COVID. Okay, this Covishield. I got fever. So this is my body's response to the drug, but few of them may not get fever. So this is a characteristics of the individual, how your body's immune system is reacting or how your body is responding to the drug. This also is an important factor in order to, evaluate whether there is an immunogenic response or not. The next slide is only about to discuss on a typical vaccine composition, just to draw your attention on the current situation that, a vaccine not only contains an active ingredient, but it also has adjuvants, different antibiotics in order to protect it, have reduction in the bacterial growth, different stabilizers. So immunogenicity can also be caused because of all these components. So it is very essential that when you actually pack a vaccine, you have to ensure that none of these are contributing to any immunogenic effect or altering the efficacy of the drug which is being administered. Now, coming to the next slide, what is the consequence of an immunogenicity? Now, an immunogenicity consequence, the first consequence is that it will start generating antibodies against the drug, which is not our intention. Okay, the immunogenicity is an unwanted thing for a biosimilar, as I told you. So if it starts generating antibodies, we have to then evaluate it, whether it is a binding kind of antibody or a neutralizing kind of antibody. See, if it is a binding kind of antibody, it will not alter the efficacy. The reason is a binding antibody will go and bind to the drug in every site other than its active site. So the active site still remains free to go and bind to its specific target and do its function. But a neutralizing kind of antibody will go and bind to the active site and thus will block the efficacy of the drug. So even though there are few drugs which are found to be immunogenic, they are still working well in the market. The reason is these drugs are found to have antibodies which are of binding type. Okay. We also can see antibody responses against hosts, hosts proteins, which are known as host cell protein antibodies, HCP antibodies. For example, if a particular protein is being expressed in an E. coli and there are certain sequences that comes in, these sequences are being recognized as foreign sequences by the body, and the body starts generating antibodies for them. These also will not impact on the efficacy of the drug, although the upstreaming and the downstreaming processes try to make it pure to its best possible form, but there are instances wherein we have seen that host cell proteins are carried on to the final drug product, but they are not deleterious or impacting either on the safety or on the efficacy of the drug. The next slide is talking about the different regulatory requirements which are guided by this, journals and research papers which are available in open domains of how to test a particular drug, whether it is immunogenic or not, and how to infer the data. So what is important is that you see that even if you see that the drug is immunogenic, you have to evaluate and you have to test it with validated methods to say that they are not neutralizing type but binding type. And all these regulators have come up with their regulatory requirements through which they actually speak to you on how to do these tests, what is the flow of work that has to be done in order to test them, and how to finally infer the data that you have got. The next slide actually tells you on where all the immunogenicity comes into picture. So as you can see, it is throughout the clinical trial, you have to have an immunogenicity assessment, especially for a biosimilar. You have to have it in phase 1 just to see whether the safety of the drug is of a concern because you are giving it to the healthy volunteers. Your phase 2s, wherein you have a larger population and you have more chances of evaluating the safety in case you have missed or skipped some of them in the phase 1 because of the smaller population. And of course in phase 3, wherein both efficacy and safety plays an important part. Also, immunogenicity plays a very important role in post-marketing surveillances, which is phase 4 clinical trials, wherein you have to actually evaluate whether the drug is safer in its longer run. So immunogenicity is a very important aspect of this clinical trial and evaluating the safety of a drug. Just giving you a nutshell of how exactly an immunogenicity data is being evaluated. It may look very complex to you, but I'll try to simplify it so that you can understand in a layman's way. So immunogenicity basically is our quasi-quantitative method, because they don't give you a exact value. The reason is that for an immunogenicity assay, the controls that you use are not raised in the same species. It is coming from a different species. For example, if you are doing an immunogenicity assessment for insulin for a clinical trial, the positive control you are using is generated in rat or rabbit. So human antibody and rat antibody cannot be compared. It is not apples to apples. So it is always a quasi-quantitative method. And these regulatory bodies have come together to, formalize a method or a workflow through which you can actually come to a conclusion which is globally accepted. So when you take the sample, when you receive the sample for your safety assessment, the first test what you do is to say a yes or no. So in the first test, you actually say whether you have antibody or you don't have antibody. It's a very straightforward test wherein you take the sample, put it into analysis, and these analyses are generally being done by ELISA-based assays, radioisotope-based assays. There are new high-end techniques like MSD, gyros, and alpha ELISA, which are more high throughput and they generate more amount of data in less amount of time. But the principle remains the same. It's all enzyme substrate reactions, or mostly protein-protein interactions. So the first test you say in an immunogenicity assay is yes or no, and that is called as a screening test. If it is no, the story closes there. If it is yes, you then say whether it is a true yes or a false yes. Now, there is a much more complex method involved in evaluating how to say whether it is a true yes and a false yes, but I'll not go into the detail. For you to know, I'm just giving you an idea that, when you say that in the first test you have said yes, there is antibody. In the second test, you have to figure out and say that whatever I have declared in my first test is a true result and not a false result. So that is what you do in a confirmatory test. Once you have done that testing in confirmatory, that yes, whatever I have reported in my screening test as positive has come true in my confirmatory, I go and do a neutralizing antibody test, which is a functional assay. You put it in live cells, which has which has shown a specific functionality against that particular disease, and there you determine whether it is a binding kind of an antibody or a neutralizing kind of an antibody. All these documents that you are kind of generating during these all testing are consolidated in a bioanalytical study report, which is called as BACSR. This goes along with the clinical trial report as a part of your safety and efficacy evaluation data. And this is what a FDA or an EMA comes and checks. So you have worked out for almost 10 years to bring a molecule to a clinical trial phase. You have done lots of upstreaming, you've done a lot of downstreaming, you have worked on different scale-ups and different processes through which you can get a better purity of the molecule, have tested different formulation. But what ends the game is the bioanalytical report. If the drug is found to be equally effective as compared to a marketed drug or better, then only the drug gets into the market. Otherwise, these drugs are being chopped out. So that is where it is very important to to have a proper, a very strict safety and efficacy data for each drug in order to declare the drug that is safe for its human consumption. The subsequent slides are more on, how exactly what are the challenges that you have during an immunogenicity assessment. So I don't think that is required at this point of time. Dr. Sangeeta, we can go to slide number 36, which is, a nutshell of what exactly we figure it out during an immunogenicity trial. So, what we do in immunogenicity assessment is to see whether the protein, the biotherapeutic, what is being given is safe for its human use. if there is a antibody expression that is being found, whether it is a binding or a neutralizing kind of antibody, and also we evaluate whether the antibodies that are being produced are altering the efficacy or not. So if it is a neutralizing kind of antibody, it will definitely impact on the efficacy. It will not allow the drug to go and bind to its receptor, and maybe the subsequent impacts of the drug on the disease will not be available. So all these parameters are being evaluated during safety assessment, and these are all interlinked with each other. With this, I go to my last slide, and I thank you all for your patience hearing, and I am open for questions.
Anybody has questions?
Sir, I'm Kokila, a faculty in biochemistry department. I just have one small, my curiosity. I mean, for my clarity, I want to ask this question. Like, as we know, for the SARS-CoV-2, we have so many different types of vaccines, which are like mRNA vaccine, the Pfizer or Moderna, or the Covishield, like Covaxin, which we are taking it, inactivated vaccines. And Sputnik is also a viral vaccine. My doubt was like, when Sputnik vaccine is taken into India, like where we are allowed, that looks like a phase 4 trial. And before even going to the population level is like again they do the phase 1 trials, take a subset of population here and then try and then go to the larger population, is something like that?
See, okay, this is a very different scenario when it comes to this COVID vaccine. You all should understand this COVID vaccine is an emergency vaccine. Okay, it did not have that luxury of time to, have an extensive study. Okay, so the kind of timelines or the kind of approaches that these vaccines are being taken, considered in order to bring these vaccines to market is very different from traditional or a standard protocol which is followed for other vaccines. Okay, so what happened is that for Sputnik, what I understand is that there is an extensive trial being done in Russia. Okay, the population is being evaluated now. While they are marketed, the first marketing will happen based on that data. So that data is being reviewed by the Indian Medical Council, and based on the safety and efficacy evaluated with that clinical trial, the drug will be marketed in India, because we really don't have time to do a full-fledged clinical trial, phase 3 clinical trial across India, across different population in order to see its efficacy. Maybe it will happen parallelly. It is still happening in parallel. And it's a continuous process. This virus is mutating every single day, and the more it is replicating, the more mutations are being observed. So there are a lot of scopes where we have to evaluate whether this particular vaccine which we are giving is effective on that particular mutant. So there are a lot of scope of research for the SARS-CoV vaccines. But to answer your question, yes, all these vaccines which are being given to you have undergone a particular clinical trial in some country. It has a data, and the data is being evaluated, and post-evaluation only it is being allowed to be given to population. So it is absolutely safe to go for these vaccines for now, because that is the only shield that you have against this virus.
Thank you, sir. So there is one more question in the chat box.
Yeah.
This is from a student. She is curious to know, sir, I have come across a new statement stating fake remdesivir's cure patient, and they say it's due to placebo effect. Is it just psychological, or does it have anything to do with placebo being a sugar as 2 DG is 1,2?
No. So first thing, 2DG drug that the DRDO has manufactured is not a placebo drug. Okay, please don't confuse it with the placebo drug. It has a different route of a different mechanism of action, and it is well established. So that is a different thing. A placebo ideally cannot cure a patient. So and also remdesivir is not a drug against COVID. Okay, there is no drug against COVID as on today. What vaccines you are getting is just preparing your body to fight against the drug. Okay, all these patients who has been given remdesivir or ivermectin or all these drugs which are coming in or in news and in buzz is to treat the symptomatic treatments. Okay, most of these COVID patients whosoever has been treated are being treated with B complexes, zinc, which only boosts your immunity. These medications which are actually being prescribed worldwide is only to boost up your immunity. These drugs like remdesivir and ivermectin are given in special cases wherein there are symptomatic. These are kind of symptomatic treatments. There are symptoms being observed which are found to be treated with administration of remdesivir or ivermectin, and that is why they are being given. So remdesivir and ivermectins, these are not drugs against COVID. There is no drug against COVID as of now. We only have vaccines which help us in protecting ourselves from future infection, because our bodies are made to generate antibodies which are stored in our bodies as well as in the body's memory cells, so that when there is a future infection, you can start preparing those antibodies and your body can combat. Placebos generally don't cure disease.
Yes, sir. Thank you, sir. One more question by student Neha. Sir, does bioinformatics play an important role in clinical studies?
Yes. So this is a very good question. Bioinformatics may not play a direct role in clinical studies, but it actually plays a very important role in preclinics when this drug is in discovery and development phase. So through bioinformatics, there are various modeling studies that are being done through which the structure of the drug is being, in silico created. Bioinformatics is, using different softwares in order to create the structure of the drug, try to figure out how best the drug can be modulated in order to to make it more effective for its intended purpose. So accordingly, the gene structures, the gene that are being impregnated in the vectors can be designed. So all these studies, all these bioinformatic work finds its use mostly in the preclinics. Also, nowadays, bioinformatics is widely used for detecting genotoxicity. Whether the drug will cause any toxic effect in a genetic level can also be figured out by bioinformatics. There are various tools which are available. There's something called Derek analysis and SARA analysis, with which they do this kind of genotoxic assessments. So yes, bioinformatics plays a very important role during the early phases of drug development.
So there is one more question from our guest, Jayant. Is vaccine permanent, or do we need booster doses, or does it has any certain time limit in our body?
Yes, so vaccines are, so you need to have booster doses in order to keep your body awake and triggered of this particular kind of infection. So booster doses actually helps you in order to elicit the antibodies, keep the antibodies awake in the body. There are few vaccines, against which the population has been resistant. So, for example, you may have got during your childhood a vaccine against tuberculosis or a vaccine against diphtheria. Those diseases does not exist anymore. So you need not have to have boosters for them. But for a vaccine like COVID, each day it is changing its structure. So you need to keep your immune system awake. So that is the reason these vaccines are being suggested to have continuous boosters in order to keep your immune system triggered and awake so that they can fight on this virus.
So there is one more question. Post-COVID, after how many days we should be vaccinated?
That's a difficult question. So
I think
that's a difficult. Yeah, I think that's what Dr. Sangeeta said rightly. I mean, as it is changing and each time, the research, as I said, is ongoing. So as we are being vaccinated, there are parallel studies that are being going in population between 12 to 18 years, whether these vaccines are safe for treatment of those young population, So and also you have to understand that this strain is mutating. So the efficacy is being evaluated, as and when there is a mutation coming in, you are testing the strain, whether it is actually impacting them. So there are changes in the manufacturing process, or there are changes in the genetic structure that these vaccines are being manufactured on. So you have to get vaccinated as long as this virus is existing, is what I understand.
Thank you, sir. Over to you, Ivava Prabha, ma'am.
Yeah. Thank you, sir. I'm sure that all of us have been able to enrich the territory of our knowledge and would definitely have an innovative notion towards the subject with enormous interest. Sir, your talk and overview of clinical trials was very inspiring, and I'm sure that the students have learned something new. Thank you, sir. We have arrived at the end of day 1 session. However, our hopes are held high to look forward to such conferences or gathering in the upcoming days, providing impetus to our innovative minds. I would once again like to thank Dr. Ganesh Ramachandran and Mr. Somnath Mukherjee for readily accepting our invitation, and I certainly look forward to hear a lot from you in the near future. Firstly, I would like to take this opportunity to thank the management and chairman of the Oxford Group of Institutions for their constant support. We have been very fortunate enough to have our dynamic principal, Dr. R. Kavyashree, to whom I would like to express a heartfelt gratitude for her and her guidance. Department of Biochemistry would like to express a huge degree of gratitude to both of you, sir, for being with us today, even amidst your busy schedule. We are highly grateful to you and would be much delighted to have much more associations in near future. I express my sincere gratitude to Vice Principals Dr. Bharati S. and Professor Gayatri Sudhir for their encouragement and support. I must mention a deep sense of appreciation and regard to Dr. Sangeeta Roy, Head Department of Biochemistry, for her constant support and energetic encouragement in all the endeavor of our departmental programs. A special thanks to Mrs. Ujwala Raju, Management Representative of HSR Campus, for her motivation. I would like to express my sincere thanks to all the participants who have virtually joined us today to gain knowledge, become more aware to current situation. The organizing team of this program, which includes the very motivating and dedicating faculty of the Department of Biochemistry, are highly regarded for their enormous efforts and support in the meticulous planning for smooth execution of this event. And on this platform, I would like to thank each and everyone. Last but not the least, I would like to express my wholehearted thanks to Mr. Prakash, System Administrator, for his constant technical assistance and for providing extent coverage of the program. Thank you, sir. Feedback link has been posted on the chat box, and I request all the participants to fill it up and let us know your thoughts about the program. Signing off today's day 1 session, and we all will meet in the same platform on Monday, that is 7th June 2021 at 10:30. Thank you one and all.
Thank you.
Thank you, Somnath. Thank you, Ganesh.